My name is DrMauricePEng. I'm a professor of visual neuroscience at the University of Montreal, and I'm also the holder of the Harlan Sanders Chair in Visual Neuroscience. My research interests concern mainly the development and plasticity of the brain, and with an emphasis on the visual system. I've been interested for the past 30 years in finding the neural mechanisms that subtend this plasticity. Two lines of work are of importance. One concerns my studies in anatomy and with animals, and the other one my studies with humans that have been suffering from blindness at birth. The first line of research indicated that the brain is indeed plastic, meaning that it changes following many interventions, and we believe that the brain is capable of rewiring itself. We showed, for example, that the retina that sends its inputs to the visual cortex can be rewired to send its input to the auditory cortex, and the auditory cortex therefore becomes responsive to visual inputs. These basic results triggered a new line of studies using up-to-date methodologies like functional magnetic resonance or positron emission tomography. We decided, therefore, to investigate the absence of visual input since birth in humans and their effects on the development of the visual system and therefore the brain. We found out that blind individuals could recruit their visual cortex when they were confronted with tactile or auditory tasks. We used a system that we call the tongue display unit, which is transducer of visual inputs into electrotactile stimulations. This system, called the tongue display unit, has been developed by late Professor Paul Baccherita from the University of Wisconsin. This system is composed of a grid made out of 144 electrodes. The grid is applied to the tongue, and a camera converts an image into pulses that are delivered through the grid to the tongue. The consequence of this stimulation with training makes it that the blind can recognize the stimulus, so the visual object, and to do so they will recruit their visual cortex. This is an extremely interesting phenomenon since it supports the concept of sensory substitution, meaning the substitution of one sense by another one. So in our case, the lost sense, which is vision, is replaced by touch. The question is how this comes about, this sensory substitution. And that calls for the notion of cross-modal plasticity, meaning that the visual cortex will be substituted by another cortex, in our case touch. Now that means that touch will acquire some of the specific components of the visual cortex that is deprived. To our big surprise, if one takes metabolic scans at rest in the blind, one will find an increase in the metabolism of the visual cortex. So not only the visual cortex is present in the blind, but it is also suprametabolic compared to normals. Moreover, we have found that the thickness of the cortex, of the visual cortex, is bigger in the blind and in controls. And the third thing is that the volume of that cortex is reduced by about 20%. And this is a very puzzling results that is also increased by the fact that many of the projections between the cortices are increased in the blind. Like, for example, the parietal cortex and the occipital cortex are connected by fasciculus that is bigger. So now we find ourselves with a rewired blind brain. What is the nature of this visual cortex in the brain? It is not by no means visual, since these patients have never seen. So we believe that this cortex is an extension of the remaining cortices that did not undergo the developmental pruning usually happening in normal individuals. That means that there would be a bigger number of synapses that have not gone away during development. So the process of elimination was completely disrupted during development. We believe also that there would be a big drop in the concentration of the neurotransmitter GABA within layer 4 of the occipital cortex of this individual that could explain the increase in the activation at rest. Our work has led to many beneficial things. Like, for example, we are awarded the Ireland Standard Chair in Visual Science with an amount of $1.2 million. The Danish government have put forth about another $1 million, plus, of course, the various agencies in Canada and Quebec. We are now carrying on this project in collaboration with Denmark. Actually, the two centers in Denmark, the Danish Research Center for Magnetic Resonance and the Department of Neuroscience at the Panum Institute at the University of Copenhagen, have been contributors to these projects since the beginning. So we have students now that who work in the different centers, and it makes a very beneficial exchange between them. I was, for this work, I was awarded several honors. In Denmark, I received the Hansen Prize for excellence and the contribution to well-being of humans. And I was made a member of the Royal Society of Denmark. I'm also a member of the Royal Society of Canada and the recipient of the William Dawson Prize of Excellence.